times. On slide 2, we're marching through the different structures or mixtures that we're going to have to encounter if we are following the pathway of light. Light has to pass through the cornea, and even before the cornea, it's got to pass through skin, but very thin, transparent skin. Then it has to pass through the aqueous humor, then through the lens, then through the vitreous humor, then through the bulk of the neural layer of the retina, and finally encounter really the posterior most portion of photoreceptors, rods and cones, okay? And along that pathway, light gets bent or refracted 3 times. Upon entering the cornea, upon entering the lens, and upon leaving the lens. And most of that bending actually occurs upon entering the cornea, and that's not adjustable. We don't change the thickness or the shape of the cornea. We do, however, have the mechanisms in place thanks to ciliary bodies, which are really muscle, excuse me, and ciliary zonules, the tethers between ciliary bodies and the lens to change the shape of the lens. And so the refraction upon entering and leaving the lens is adjustable, okay? Slide number 3. In a normal eye, we like our lens, we prefer our lens to be, why can't I draw? That's weird. Huh. Oh, maybe my pencil's dead. We'll plug that in just in case. Anywho, we like our lens to be stretched out or flattened comparatively for distant vision. And distant vision is overseen by the sympathetic nervous system. We achieve that flattening of the lens by, I always have my body to use as a demo for this. I don't have that right now. We'll see if I can explain this successfully. The ciliary bodies, or body really, which is also a muscle. So ciliary muscle, ciliary body, same thing. It's allowed to relax. And when it relaxes, it slumps. It weighs down. Kind of like a marionette that is magic, of course, as all marionettes must be, and asleep. If it were sleeping, otherwise lively marionette, it would hang, it would slump on its strings, okay? And by that same principle, a relaxed ciliary body will slump, hang, pull on ciliary zonules and keep them taut. And by pulling on the ciliary zonules, we're pulling on really the rim of the lens and flattening it out, stretching it out. Very, very interesting stuff. Let's look at, we're going to go to slide 5 next because we're skipping slide 4. Just in the interest of time, but also because this quarter is so strange, I want to keep things as basic as possible. Anywho, if we want to focus for close vision or near vision, well, that's overseen by the parasympathetic nervous system. And this time around, we want that lens to bulge. We want it to be relatively thick, okay? And in order to achieve that, what we're going to do is we're going to wake that magic marionette up. And so being magic and lively, it stands up, and the strings that are attached to it are now nice and loose. In other words, the ciliary muscle or body contracts or stands up and allows the ciliary zonules to be quite loose, and therefore allows the lens to bulge. Okay. Slide 6. We're skipping this quarter, even though it really bothers me to skip it. Slide 7, slide 8, we're skipping this quarter. Slide 9, excuse me. Photoreceptors, again, are neurons. Any kind of receptor is a neuron that are sensitive to changes in light. There are 2 types of photoreceptors in the retina, the rods and cones, okay? And each photoreceptor consists of some predictable parts and some not so predictable parts. Each one will have a cell body, a synaptic terminal, and 2 segments, an outer segment and an inner segment. The outer segment is actually the portion of the photoreceptor that is stimulated by light. It's the portion that light is headed toward as it moves along its pathway. And that outer segment looks a bit like an earthworm because it's subdivided into discs, and so it ends up looking like a segmented worm. I think it also looks either alarmingly or delightfully akin to the thylakoid membranes in chloroplast, which actually serve the same general function of being sort of photoreactive. So it almost looks like we have chloroplast in our retina, which is just fascinating. The outer segment also houses photopigments, yes, just as chloroplasts do. And those photopigments are molecules or proteins that change shape when they absorb light, okay? And that really starts the cascade that will ultimately be perceived as vision, okay? The inner segment just acts as a bridge. It links outer segment to cell body, okay? Now, photoreceptors, they are damageable. We actually damage them all the time. And they're more rapidly damaged if we suffer an injury that maybe detaches the retina. They're more rapidly damaged if we expose our eyes to intense light. But even if we don't go out of our way or have an accident to damage our photoreceptors, they will be damaged. And the good news is that we're actually constantly replacing them. The entire outer segment in particular gets replaced about every 24 hours, okay? And then those older, really, discs of the outer segment, they're phagocytosed by that pigmented layer of the retina, those cells that look kind of like macrophages and have so much surface area available. Will phagocytose older discs of outer segment. Okay, let's go to slide 10, which lays out, let's see if this pencil's working now. Yay! Lays out at least part of the retina, okay, where here is one of those surface area rich cells that make up the single cell layer that we call the pigmented layer of the retina, okay? And then everything else that we're seeing here is neural layer, although we can't see all of the neural layer. Up here we're getting a peekaboo of the bipolar cells, okay? Light is coming in from this direction, or really because of the way that the illustrator is showing us, or orienting his eye, I should say. It's coming in from this direction, but whatever. Bipolar cells are synapsing with photoreceptors. These guys are the photoreceptors. Okay, and notice that much of the outer segment is actually embedded in those phagocytosing pigmented cells of the retina. So this is an outer segment, rod-shaped, therefore that's a rod. This is an outer segment, cone-shaped, therefore that's a cone. This is an outer segment of a rod. This is an outer segment of a rod. If you look really carefully, you can see that these outer segments are divided into discs. They almost look like they're ribbed. Okay. Well, the oldest discs, so hard to say, are here. And the newest discs are here. Okay? And in fact, these regions of this particular photoreceptor's outer segment are being phagocytosed. Because we're constantly making new, we're also constantly degrading old. Okay? Here's the inner segment, just a bridge really between the soma and the outer segment. Here's the soma or cell body, okay? And then here are synaptic or axon terminals. I don't care, either one's fine. All right. And then also in this little cutaway, you can better see, here are the bipolar cells, here are the ganglion cells, here are the photoreceptors, okay? And then here would be vitreous humor. Slide number 11. We've already started comparing rods and cones, but ultimately you want to be able to compare them across a multitude of points. Again, rods are more sensitive to light, and therefore we don't need a lot of light for them to fire. So they're best for low light level vision, night vision, okay? They're best for peripheral vision. And rods only feature one type, one flavor, if you will, of photopigment. And that's why they don't produce, if you will, color images. They only function in producing like grayscale images, okay? The pathways or neurons of rods, they tend to converge ultimately on ganglion cells, and we can see as many as 100 rods converging ultimately on one ganglion cell, and that's why rods generate fuzzy, indistinct, poorly resolved images, whereas cones, they have low sensitivity to light, sensitivity, there we go. And therefore we don't need or rather they won't fire in poor light. We do need bright light in order for cones to be activated. They do respond more quickly than rods do. They have, each one can have, one of 3 different flavors of photopigments. So cones, they come in 3 flavors, 3 different photopigments. And it's those 3 different photopigments and sort of their overlap and blend that actually generate tons and tons and tons and tons of different colors for us, or at least generate the perception of tons of different colors, okay? Fewer cones will converge on ultimately ganglion cells, and that's why cones are much more so able to produce resolved, distinct, very clear images, or at least in our perception. Let's go to slide number 12. Slide number 12 does a side-by-side comparison of rods and cones. I would think ultimately you'd actually be able to generate a larger table than this, but at least it sort of starts that ball rolling. Slide 13, visual pigments or photopigments, same thing. Retinal, not to be confused with retinol, that is featured in some beauty products. I almost said beauty projects. Retinol is a molecule that kind of forms the base or basis or foundation for any photopigment in the retina. So if it's the photopigment of a rod, yeah, retinol is there. If it's one of the three photopigments that we can find in a cone, then yeah, retinol is present, okay? And in order to create different kinds of photopigments, one flavor for rods and 3 other flavors for cones, retinol has to combine with molecules that do differ. And those molecules are called opsins. Okay? And all of this is protein, just proteins that combine with other proteins, okay? Opsins are, I guess you could say they're the component that conveys color, okay? Retinol is derived from or synthesized from vitamin A, so no wonder, we want to store vitamin A in that pigmented layer of the retina, okay? Again, there are 4 different opsins, right? The opsin that's found in rods, in the outer segment of rods, is called rhodopsin. And I've got some really bad, really frustrating news for you, okay? Yeah. The rod photopigment is a combination of retinol and this opsin called rhodopsin. Okay? But often the combination of the two is unfortunately also referred to as rhodopsin, which is really, really frustrating. It's kind of like saying pencil plus lead equals pencil. Gross. Now, there are 3 other opsins, of course, those that are found in cones, and those have easy peasy names. Green opsin, blue opsin, red opsin, and they're really named for the wavelength of light that they absorb or that they're sensitive to, okay? There is some overlap in the wavelengths that trigger these different cone opsins, and that overlap is actually chiefly responsible for the huge variety of colors that we perceive, okay? Let's move to slide number 14. Retinol can be found in 2 different sort of shapes or forms. If it's bent, then it's called cis retinol. If it's straight, then it's called trans retinol. And it turns out that retinol takes its bent form in the dark. Okay, when exposed to light, it takes on its trans form, right? And we can convert retinol from its cis to its trans, from its trans to its cis, quite readily, and we do that every day, all right? Depending on the shape of retinol, we may or may not allow retinol and opsins to combine and act as photopigments. Slide number 15. So at the top of the slide, here are some of those discs in, for instance, the outer segment of a rod. And often we use rods as our model, but cones really work the same way. Okay, and embedded in the membranes of those discs, we can find lots and lots of different proteins, but just one of those proteins is an opsin. If this is a rod as our model, then this particular opsin is rhodopsin, okay? And it is near or in conjunction with, sorry, these pointers don't align the way they originally did. With this guy, that's retinol, okay? They're nearby each other all the time, but it's really a question of whether they're bound to each other, that they will form a complete photopigment, unfortunately often referred to as rhodopsin. Let's look at slide 16. Okay. Rhodopsin, if we're using rods as our model, they need to capture light, and they can only capture light if we're talking about a fully formed rhodopsin, not just the opsin, but the photopigment. Okay. And there are sort of three different steps, and in what is actually going to turn out to be a cycle: pigment synthesis, pigment bleaching, and pigment regeneration. Slide 17. We generate that photopigment, we synthesize that photopigment when opsin, in this case a rhodopsin, okay, and the cis form of retinol combine, okay? And that happens, of course, in the dark. How can I tell? Well, cis is the dark form of retinol, okay? When that complete photopigment absorbs light, the cis form of retinol will change into the trans form. And, excuse me, the trans form does not have an affinity for that opsin, and so they separate. All right, so pigment bleaching and pigment breakdown, same thing, okay? When we are exposed to dark again, or rather when we're no longer exposed to light, that trans form of retinol will be converted back into the cis form, ready to do it all over again. Okay, let's go to slide 18. This image is really scary, in part because it's so wordy, but don't worry, I'm going to walk you through and everything's going to be fine. In the dark, our retinol molecule is in the cis form, and cis retinol does have an affinity for opsin. Therefore, in the dark, we have a complete photopigment. When that photopigment is exposed to light, that cis form gets converted into the straight trans form. The trans retinol abandons the opsin. They're still very close to each other. They still have access to each other. They just don't have a high affinity for each other. Okay? And then when we are exposed to dark again, or we remove that light, we can reform cis retinol, which again does have an affinity for opsin and therefore regenerate, reuse, cycle through. Okay? Slide 19, please. Excuse me. When rhodopsin, I'll say photopigment. When the photopigment is exposed to light, and that cis form gets transformed into the trans form, and therefore the trans form of retinol abandons the opsin, that actually starts a whole cascade of reactions, a molecular relay race, or a metabotropic pathway, okay? In the dark, when we're not exposed to light, and those photopigments are whole, they are complete, they are formed, we hold ion channels, specifically sodium and calcium ion channels, that are in the membrane of the outer segment of a photoreceptor open, okay? And therefore, in the dark, a photoreceptor is in a depolarized state, okay? Whereas when we're exposed to light, those channels are forced closed. Those same ion channels are held closed, and therefore photoreceptors are held in a hyperpolarized state. Okay? Well, if you remember action potentials, and oh my goodness, you better, then you remember that a neuron is firing during depolarization. It is headed back down toward resting during hyperpolarization, right? And you might think, well, vision occurs when neurons are firing. Nope. Vision is actually triggered. The cascade that leads to vision is actually triggered by the hyperpolarization of photoreceptors. Isn't that interesting and counterintuitive? So cool. Let's go to slide 20. Photoreceptors are very short neurons. Bipolar cells are very short neurons. They're short enough that they can afford to only generate graded potentials. They don't generate active potentials. Okay? And so that means that they can be either stimulatory, their impulse can be stimulatory, or inhibitory. Okay? When we're exposed to light and a photoreceptor hyperpolarizes, that photoreceptor stops releasing inhibitory neurotransmitter. Therefore, stops inhibiting bipolar cells. That's misspelled, and I've only just noticed it after years. That's funny. Okay? That bipolar cell is therefore free to act. It's no longer inhibited, and so in the light, it is allowed to depolarize. Right? And when bipolar cells depolarize, they release excitatory neurotransmitters influencing ganglion cells. So cool. Ganglion cells are longer and therefore do generate action potentials, which travel along the optic nerve. Slide 21, please. All right, here we are in the dark, and photopigments are intact. In other words, opsin and cis retinol are combined. They have an affinity for each other, and they are therefore forcing. Here's another typo that I'm only recognizing for the first time ever. Channels. They're holding cation channels open. Okay? Those cations, therefore, are going to flow into the photoreceptor and depolarize that photoreceptor. And again, we're in the dark. Okay? That photoreceptor is going to generate an inhibitory graded potential, and therefore it is going to secrete inhibitory neurotransmitter. It releases that inhibitory neurotransmitter into the synaptic cleft between it and its associated bipolar cell, and therefore inhibits the action of that bipolar cell. Okay? Bipolar cell is not going to release its neurotransmitter, and therefore it is not going to generate an excitatory action potential, which is a little redundant. All action potentials are excitatory in a ganglion cell. So no vision, no sight, if you will. Let's look at slide 22. Really the same layout, but now different conditions, okay? We're exposed to light, therefore our photopigment dissociates, our trans retinol abandons our opsin, okay? And that causes cation channels to close in the photoreceptor, okay? The photoreceptor hyperpolarizes and finally, frankly, shuts up. It no longer is releasing an inhibitory neurotransmitter onto bipolar cells, which allows bipolar cells to finally release their neurotransmitter, which is excitatory. That release of excitatory neurotransmitter causes or initiates an action potential in ganglion cell, and voila, vision. So cool. Sorry about all the background noises.